Good morning. I'm Paul Neam from Dermatology, and it's my real pleasure this morning to introduce this year's Odland lecturer, Tony Oro, coming to us from Stanford. George Odland, for whom this lectureship is named, was the division head for dermatology for over 2 decades, from the 1960s to right around 1990. Grew the division very impressively and described basic structures and functions of the skin. Tony is the professor of dermatology and associate director of the Center for Definitive and Curative Medicine, a member of the Institute for Stem Cell Biology and Regenerative Medicine at Stanford and at the Stanford Cancer Institute at Stanford. He did his training, MD, PhD, with Ron Evans at the Salk Institute, and then did a postdoc with, after he did his dermatology training, with Matt Scott, where he studied hedgehog signaling, stem cell biology, and the basis for our hair and basal cell carcinoma, the most common type of cancer in humans. He then, as a physician scientist, led clinical trials that characterized the first inhibitors of the hedgehog pathway, so-called smoothened inhibitors that are therapeutic for more aggressive basal cell carcinomas, and has a long-standing interest in that area as well. And tomorrow he's going to be talking to our dermatology group about the basis of resistance to those drugs in basal cell carcinoma. But today he's talking about something completely different, relates to his fundamental interest in stem cell biology and human skin development. And he is now leading a program in therapeutic reprogramming. As he refers to it, this is like training cells to behave. It's like training your pets to behave and go into an organ and persistently behave in a way that you want. It is not trivial. They are evolving. They are alive. And he's going to tell us some of those battles that he's dealing with today, including treating perhaps the nastiest skin disease of all, epidermolysis bullosa, which is a collection of genetic diseases where the epidermis does not connect to the dermis properly. And you can imagine your epidermis is sloughing off, you are in big trouble. Usually that's one tiny mutation in a protein. It seems like a trivial problem to address. Tony's going to tell us the progress in that area and the huge challenges as well. Tony, thank you very much for being our Odland lecturer for this year.
Thank you. Thank you very much, Paul. Can everyone hear me in the back? Great. So I'm very honored to be the Odland lecturer. I think George Odland was a pioneer in skin biology. And I just, for the audience today, I really wanted to just tell you that and try to convince you that dermatology is a great specialty to do research in. It's a wonderful model for disease, mirror into disease processes. And in terms of the ability to take and look at skin, throw it in a dish, manipulate it, and then understand it in terms of, as a pioneer for cell therapies and small molecule therapies, really is a great model for translational biology. And so I hope by the end of today, you'll see that it really is a way to move forward therapeutically on the skin. So first disclosures, actually none that are actually particularly relevant for today's. So what I want to talk about today is our cell therapies. And so this is medicine grand rounds. If you were actually going to go to surgical grand rounds, the first thing they would ask any of the residents that are here would say is, how do you cut it out? Okay, so if something's bad, you have a gallbladder, but you hack it out. Now here in medicine grand rounds, you ask which drugs you give them. So it's an antibiotic, you have an infection, you get pimped about which antibiotic and resistance and why you want to preserve antibiotics. But what happens then if there's not something you can cut out, not something that you can give a drug to, but the tissue, is this still on? Good. But the tissue that you've been actually diseased, you can't cut it out, and you can't give a drug to it. What do? And this is the issue. This is a huge hole in our armamentarium because you can't cut it out and you can't give a drug to it. So what do you do with it? So, for example, if your skin is defective, you're born with a genetic disease where your skin falls off, you can't cut it out and you can't give a drug to it. So what do? You're basically messed up. You're stuck. There's nothing in our armamentarium that can actually fix that. And that's the hole we're trying to fix. And that's the case for actually a surprising spectrum of diseases, which we want to talk about today, where we have nothing to offer our patients. That includes monogenic skin disorders, for example, or monogenic disorders in general, epidermolysis bullosa here on the left, which again, as Paul had mentioned, is a monogenic skin disorder where there's one of 19 proteins which connects your epidermis to your dermis, and if you're happening missing one of them when you go through the birth canal, which is a very traumatic place, it comes right off, and then you're basically like a burn victim for your entire life. Also, more ones you've heard about: hemophilia, immunodeficiency disease. These are diseases and other disorders which we really can't fix. Okay, so those are the rare monogenic disorders. What about common diseases with genetic components? For example, one of my favorites, male pattern baldness, which affects many of us, including me in the room. Very important genetic disease with a common disease with a fraction of a genetic component. Neurodegenerative disorders, which are now increasingly understood to have are common diseases with large genetic components. Diabetes is another example. And then even more common replacement tissues. So I'm a runner. After years of running downhill, my knee cartilage is looking pretty sad. And so what do? Back, and then for skin burns. We can't replace it. So here's an example where we need regenerative medicine. We need actually to replace the tissue at the time of the disease. And this is an example of a axolotl where you can cut off the limb at a certain time, and the stump actually then induces embryonic tissue, has all the information it requires to pattern that organ again, and then reforms it, as you can see, with time. So can we harness the endogenous regenerative abilities that are seen in lower animals to regenerate tissues in the body? Okay, and these therapies and the possibility of these therapies working really come from this confluence, this time in history which we have, where stem cell biology and cellular reprogramming, we can take somatic tissues, make them go all the way back to embryonic-like cells called induced pluripotent cells. This is the Nobel Prize from Yamanaka, where 4 transcription factors can take a somatic cell back to an embryonic-like state, where it can then make anything, any tissue, and it has the instructions to make any tissue in the body. Genome editing technology, where you actually can then go in and fix in the stem cell itself that particular mutation. When we started this program 10 years ago, they told us you couldn't fix human genetic material. It was impossible. And now the answer is not can you fix it with genome editing technology such as CRISPR-Cas9 or AAV recombinant genetic adeno-associated virus, which I'll talk to you about. But now the question is, which one should you use? So that's where the technology is now. And then finally, advanced tissue manufacturing, bioprinting, and other types of engineering, which allows the combination of regenerating tissues, correcting the mutations, and then manufacturing the organ are now possible. Can we make them go forward? Okay, so that's the center that Paul talked about, that we're basically trying to do this at Stanford. And it really is, again, the program is a combination of tissue-specific genetic diseases, metabolic diseases, and immunodeficiencies, as well as minimally manipulated bone marrow issues like Tregs or CAR T cells, which we're trying to move forward for cancer as well. And it really is a discovery arm to spin out therapies. Okay. Really the issue is it's kind of like we're trying to be a Home Depot for preclinical medicine. If you have a therapy, we're trying to then take it and move it onto our patients. And that's really this GMP facility that's built. It's a cell therapy facility at Stanford that's headed by David Augusto, really helps us to move and develop cell therapies into the clinic. Okay, so Paul talked about this, and I mentioned it before. 12, actually now there's 19 proteins that actually link here, that link the epidermis up here with the dermis. Very fragile group of linker chains that keep this thing together. It's amazing. There's these 19 proteins. That's it. Any one of the mutations, any one of these diseases, genes can cause the blistering disorder. And so what's the cost? So one of the mildest forms is called epidermolysis bullosa simplex. It's a rare disorder, but the standard of care is really supportive. It's wrapping these kids like mummies and then keeping them from being infected and trying to give them nutritional support, and that's it. So there's really nothing else that works. And the cost just of the wound care dressings are $120,000 a year. And this is for the mildest version of this disorder. And so really the question in terms of insurance companies, people say, oh, these cell-based therapies are so expensive. But so then just in terms of cost and thinking about it as a therapy, if we can have a tissue therapy which is long-lasting and in a sense definitive, then it actually makes sense to move these things into the clinic, because when the costs, we have nothing else that works and the costs are so high just from palliative therapy, that then it may actually it actually brings the therapy down into a reasonable cost. And we can have this discussion afterwards about should we do this or not, and the cost and that sort of thing. Okay, the other thing that's important about this is that 55% of these patients with this severe disease have chronic wounding. And we know that chronic wounding accumulates mutations, and over half these patients will die from invasive squamous cell carcinoma from chronic wounding and acquired mutations. And so if we make any headway into preventing the wounding, we can then basically extend the lives of these patients. So what are the requirements for definitive therapy? So long-lived disease-modifying stem cells that will prevent future disease complications. They have to be in a transplantable tissue, a permanent correction, not something, and then also some sort of an improved safety and risk profile over the standard of care therapy. This is true for skin. This is true for bone marrow. This is true for knee cartilage. So in terms of these definitive therapies, the way we think about these things, it has to be in a transplantable tissue. So the idea for the audience is if you have a transplantable somatic setting, like for example, bone marrow transplant would be the first example, or skin grafts, corneal transplant, just think about anything like that, somatic transplantable tissue. Then these cell therapies can be for your application. So that's really what the discussion we want to have today. And I'm going to tell you about skin, but really I want you to think about for your particular disease indication whether this is actually possible. Because I think you realize that for muscular dystrophy, there's a lot of things that you don't think are somatic transplantable tissues, which actually you can if you find the right environment. For example, liver, for example, we didn't think it was transplantable, but now we know that if you wound it, you'll actually partially regenerate. So partial hepatectomy with autologous corrected liver cells actually is possible for metabolic and nutritional diseases. Okay, so what are the therapeutic approaches for this recessive dystrophic epidermolysis bullosa? These are collagen 7 mutations, which again link the epidermis with the dermis. So this is a horrible disease, and so people try whatever it takes to basically put back this matrix protein called collagen 7, and they're listed all here, and they include approaches like, can you induce a increased level of a protein which acts like collagen 7 or like keratin 14, and that's, for example, this broccoli derivative called sulforaphane, which Pierre Coulomb has been pioneering, which induces, instead of the keratin 14 mutation, induces keratin actually acts like it and actually has great results. So that's a protein mimic inducer, and believe me, many of the Epidermolysis bullosa type patients with EBS are loving broccoli. And so, which is probably one of the only people in the world that do. So you also can just produce tons of the protein because it's a matrix protein and it's like a glue. And so collagen 7 injections, and those that's being done by a number of groups, including Peter Rinkovich at Stanford and the group at University of Southern California. That's a very good approach. Some have advocated for bone marrow transplant because it's easy to do. And there's a small amount of collagen 7 that can come from the tissue, the fibroblast tissue. There's some issues with survivability of bone marrow transplant in patients that have overwhelming blistering and wound care. But what I want to talk to you today about is really about the keratinocyte program. And we chose this keratinocyte for a number of reasons. One, because it's the thing that makes about 10,000 times more collagen 7 than anything else. It's the collagen 7 factory. We also know a lot about the keratinocyte as a cell, and with work from many people here at UW about how to grow and maintain keratinocyte stem cells. In much of the work that, in terms of keratinocyte culture, have come from people here at UW, including in the audience. And so it really is a way for transfer. And so we really focused on keratinocyte in terms of either putting back the gene as a retroviral transduction, shown here, or this approach called therapeutic reprogramming, where you can actually go and go back, make the cell back into an induced pluripotent cell, correct the genetic defect, and then manufacture the tissue, or using spontaneously or induced revertance, which I'll talk about all of these. Okay, so what's the evidence that if you have a keratinocyte on a patient that's corrected, it has disease-modifying activity? This really, I think, is the critical proof of concept for cell therapy. Can you take the patient's keratinocytes, correct them, and then make little grafts, and then graft them on, and does it work? So really basic. Now you think, a lot of gene therapy is done at University of Washington, and you think this should be a very easy thing. You take some keratinocytes, you throw them in a dish, you put a retrovirus in, it expresses collagen 7, you make little grafts, you put them on a patient. So just to give you a sense, so this is a 30-day procedure which now is into the phase 2 trial. But to get this just to give you a sense of the timing, not of the proof of concept in the lab, but in terms of getting FDA approval to do the phase 1. The initial concept was actually discovered by Paul Cavari in 1997. 97, that's 20 years ago. And it took 18 years from the initial observation to actually move this in to do the phase 1 trial. 18 years. It was all done in an academic setting with money pieced together from NIH, donors. You may have seen Paul Cavari, my chair, on the street corner saying we'll make cell therapies for food. Anything you could do to get this thing to move forward, because it wasn't. And so 18 years, and that's actually how long it took to get from the initial observation to the first. So the idea for this sort of program that we're trying to move forward at Stanford is to try to shorten it from 18 years to something shorter. Before I lose all my hair and my knees are completely blown. And so this was a phase 1 trial, which was recently completed. The principal investigator was Peter Rinkovich, who discovered the collagen 7. It was done at Stanford, 4 patients, 6 wounds. It was recently published in JAMA in last fall. And the idea was to then make the graphs, showing that here in green, these patients don't have collagen 7. The keratinocytes are basically infected with the retrovirus carrying the collagen 7 gene, and now they make collagen 7. This is just to show you that it was very effective. Green, so basically 24 grafts. They're about this big. Put it on the patient, and now out 3 years, very effective in basically taking a wound up here in the upper left that never healed in their entire life, and now it actually is sticking on, and they can hit it or traumatize it, and it sticks. And so this says really that, and this, if there's the reference is down here on the right, and I recommend you read the paper because it really says if you have an autologous keratinocyte stem cell that's actually growing and has the right gene, has long-term disease-modifying activity. And we know how to produce it, we know how to culture it, we know how to, the problem is scalability, because somatic cells don't scale that well. And so, I mean, yes, burn patients, you can take a little graft keratinocytes and grow them out, but these also are pre-malignant keratinocytes, and so if you expand them out, there's issues with carcinogenesis. And so really the question is, can you improve on this therapy in some way? Because the issues are, again, low keratinocyte stem cell number in these patients with chronic wounding. Gene transfer is ineffective for dominant negative diseases. So if you have a genome editing technology that can actually revert dominance, that would be much better. Safety concerns with basically putting in a retrovirus. One of the things that shut down the program in 2003 was insertional mutagenesis next to an oncogene that caused leukemia in a bunch of patients in Europe. And so that shut the program down for about 5 years. And immunologic or biological side effects of continued expression of the defective gene, where you might get either carcinogenesis from laminin collagen 7 overexpression. And so they really can use the endogenous regulation system to do this therapy. So really, I just want to talk to you about two approaches and to get you to think about how to, maybe we might be able to do this, and why skin might be a very useful way to test a bunch of the technologies that you have here. And that would be, so the first would be a direct correction of the gene in situ. And the second would be to take out the keratinocyte, make induced pluripotent cells where you can scale them up and then manufacture as much keratinocytes as you want in vitro. So the first is this direct correction. So the idea would be then, since we know if you have a keratinocyte on your body that's corrected and it makes collagen 7, it'll have disease-modifying activity, why not just use the genome editing technologies that are now available? Don't take it out, throw it in a dish and manufacture it, just try to correct it right on the patient. This is the, as my wife would say, this is the most direct way to do it, so let's do it this way. And we have a number of different genome editing technologies that are available, including zinc finger nucleases, adeno-associated viruses, and these things, this CRISPR-Cas9 system, which basically are like surgical devices which will go in, correct the mutation without leaving a scar. And that's really the technology that's available today. And so the advantages of the adeno-associated virus approach are listed here, which is that it's already been FDA approved, the AAV virus, for gene delivery. And it's a very simple design. GMP manufacturing of AAV is actually well known and FDA approved. Very mildly immunogenic. And so this has very attractive. What we discovered, as I'll tell you about, is that there's some variants, and this is in collaboration with my neighbor, Mark Kay, who's a virologist, who actually trained and was a professor here for a while at UW. And that some variants can induce homologous recombination, can do the correction very efficiently. And so two postdocs in the lab, Sandra Mello and a postdoc in Mark Kay's lab, Leszek Lazowski, who's now in Australia, wanted to see, hey, can we develop an AAV that does homologous recombination at higher efficiency to try to then develop essentially a correction cream to wipe on to correct it, the disease in the skin. Now, you can't do this in other organs, but that's the beauty of the skin, is that can you do a direct cream to actually do the correction for monogenic disorders? Okay, so what they did, and this is some data here, is they took a number of different capsids, so the same genetic material, but then different capsid variants, because Mark Kay's lab was actually the story is that Mark and I were just sort of walking down the hall one day, and he was complaining that they're doing gene transfer AAV, and he was complaining that we got this gene transfer going really well, but the stupid virus we have integrates into the genome and actually corrects the mutation. And I'm sitting there going, really? One man's, problem is another man's solution. And so we're like, wow, that's really great. And so then we got our postdocs to work on this. And it turns out that although most people work with these variants here, these adeno-associated virus variants down below, this one particular variant, and now there's actually many other ones that we've developed, and then Mark's lab has developed, actually gives you an enormous number of recombinants here, colony numbers, compared to the rest. So this is a particular variant, a capsid variant that allows the material that's inside here, the virus, to go into the gene and then actually induce homologous recombination. So whatever you put in this adeno-associated virus, we put the wild-type sequence of the gene. It goes in and it recombines and actually will correct it, which is pretty amazing. So I'm like, Mark, what are you complaining about? Let's and that's what we did. So what we used actually for a screen was the patient cells themselves. So ordinarily mutated these Epidermolysis bullosa mutated keratinocytes, because they have a lack, in this case, this was junctional epidermolysis bullosa, which lacked an Launin alpha-3 gene. These keratinocytes, when you plate them on plastic, they don't stick. So they need to be put down on collagen to stick, but if you put them on plastic, they don't stick at all. If they have their wild-type Launin alpha-3 gene, they actually will then stick. So this is a beautiful selection. So we basically labeled the keratinocytes and then screened a wide variety of different capsids just by the number of cells that stick on a plastic plate. And so you can see that if they made the wild-type mutation here, if you correct this little stop codon, now they would make Launin alpha, the Launin 332, which is the one of the components that allows keratinocytes to stick, and it would stick to plastic. And so then we use that and use this scarless adeno-associated virus targeting construct, which is basically a hunk of wild-type 4 kb of DNA, and it basically delivers it, corrects it, and then basically see what's the frequency of the keratinocytes that stick on the plate. And just to show you the key figures right here, which is the percent of correction that's needed to allow for each different virus that we tested. And the bottom line is that really it's very efficient. But we notice is that if one cell corrects genetically, it actually then spits out Launin alpha-3 to its neighbors. And so it'll actually then correct its neighbors as well. So, which is actually a very useful thing if you're trying to, because we're not perfect, and the question is how much or how little number of cells can you correct to then actually get a lawn of cells that are surviving because there's neighbors surviving. And that's shown here. So some of the clones we picked out that stuck to the plate had 50% of the cells corrected. But actually, this was actually very interesting because down at 5% of a colony that was corrected, only 5% of the cells had a recombinant cell. So that says that really, and this particular keratinocyte must have just been spewing Launin alpha-3 out like it was like no tomorrow, and it was correcting all of its neighbors. And so really, somewhere between 5 and 50 really percent of the cells, if you can correct them, you can get a lawn of cells. That was really exciting. And even more exciting was that if you looked at the off-target recombination, which is really a critical thing, we then basically did this experiment and then took out individual clones and then sequenced their genome to see what individual clones of cells that were recombinant, how many had off-target effects, and really it was a very, very low number. We can discuss this at the end if you'd like, but so it was safe and effective and had a very low rate of random integration. And if you then take these, and I'll just walk you through this, but basically if you then take the keratinocytes, and the nice thing about human keratinocytes that are primary is that you can then make organotypic cultures that look like human skin and really ask, can you make human skin like from these primary keratinocytes? And the answer is yes. So if you take, these are the wild type cells here and the mutant cells. When you see this blister, and the green here is Launin alpha-3, which is one of the genes that is causing EB, and it's gone and it has a blister, and then you can target it and now it makes laminin alpha-3 protein very nicely and regenerates an entire epidermis. And you can see that here by immuno-EM, that this is immunoelectron microscopy to the human alpha-3 gene. Now it's making tons of alpha-3 and a beautiful hemidesmosome, the perfect glue to keep these keratinocytes on. Okay, so that's really great. So then now the question is, can you do this in vivo? So you can take the cells out and then add the adeno-associated virus, shake the cells, plate them, and the ones that stick are the recombinants, and then grow them out and make grafts. But can you then do this where you can make the cream, the AAV cream, where you can just basically hand the patient squirts it on, get recombination? Is it high enough frequency to do that? I'll just tell you the bottom line is no right now. And so this is an experiment which where in collaboration with Mark Kay's lab, where we take an adeno-associated virus that we know works in vitro, and basically the way this experiment is, we take the GAPTH gene, which is a very highly expressing gene in cells, and we try to recombine in a fluorescent protein into that site, and then you can actually see the clones that grow out in a grafted human skin. So we basically take human keratinocytes, graft them on the back of a mouse, and then inject or wipe on this virus, and then we can actually score the number of cells that are recombinant. And you can see here that you're getting recombinant cells. So we can see them and they're working. But the reason why I'm showing you the examples here is that they're just examples. That they're, what you need, again, is 5% of all the keratinocytes we estimate to actually make it work. And we're getting about probably 500-fold less frequency than that when you do it in vivo. Okay, so it's exciting, but there's still this gap in frequency which we need to close. So that's the status of the wipe-on cream. Okay, so if that doesn't work, then we need an alternative approach. And the alternative approach could be, again, is try to use therapeutic reprogramming. And so just to walk you through this is, again, this was published a couple years ago, but the idea is that for somatic keratinocytes or for other tissues which are hard to use somatically, can you then basically take your patient's own cells, use the Yamanaka factors to make them into induced pluripotent cells, correct the genetic defect, scale it up, and then manufacture particular tissues. So why would you want to do this? Why don't you just use somatic tissues? Because we all know that burn patients, you can always just expand a whole tennis court from a little biopsy. And then there's a number of reasons. One, because of the mutational burden in scaling it up. Second, because if you have a cell bank for manufacturing, then you have an infinite number of tissues you can actually manufacture from that cell bank. Also, it's defined, and you can then take out anything that's bad, and then you can then manufacture from what you have. So you have a really defined, so the idea in terms of cell therapy is you need a cell bank that's autologous and has the right genetic mutations. And so that's where therapeutic reprogramming comes in, where you can actually take the skin biopsy, reprogram into induced pluripotent cells, induce hemolysis recombination, take out the clones you don't like, pick out the clones you like, and then manufacture the end tissue, whether it's skin, bone marrow, cornea, retinal tissue for macular degeneration. We're trying it for cardiovascular tissue, post-MI as well. Joe Wu at Stanford. Maurice Vernig is trying it for post-stroke neurogenesis. So these are all possible ways to go. But the skin is actually a great starting point, because if you have a problem with the experimental tissue of the little grafts you're trying to make, and it's not going so well, you can just basically cut it out. So the FDA loves this because if you put it into the brain and you have a problem, you're in trouble. But if you have a little skin graft and that's not working, you just cut it out. So it's a nice proving ground for these novel cell therapies. Okay, so we talked about different types of genome editing technologies. I'll just mention that CRISPR-Cas9 really is emerging as a very early favorite because it doesn't require a large manufacturing ability. You can use RNA that makes CRISPR-Cas9 and the guide RNAs to actually induce the homologous recombination. And so there's some patent issues around this, which we can talk about trying to use it, but really CRISPR-Cas9 and the efficiency is much higher than for adeno-associated devices that we've seen. And so I think this is actually emerging as the technology is developing as a very efficient way of correcting or modifying the human genome. I can talk to more about that at the end if you'd like. Okay. What about, now, so if you can make the iPS cells and correct them, what about, again, getting back to this idea of a cell bank? And I keep harping on the cell bank because for manufacturing and for patients, you want to know what you start with. So a lot of the problem with stem cell biology and with reports, people say, oh, we're going to use stem cells to correct. We're going to use, platelet-rich plasma to correct your hair follicles. I mean, there's all these, reports in the literature about stem cells working. And I say most of those are actually snake oil. So how are we going to, as a field or as a physician, as a community, convince our patients that this is real and works? We need to have some big common basis for defining what we have. And in particular, the improved cell bank is important because these patients die of squamous cell carcinoma as they get older. Can you then take this cell bank and pick out the clones that have pre-malignant mutations? So the other part of my lab works basically on the mutational landscape in basal cell carcinoma, one of the keratinocyte diseases, and another person in my lab, in our department, Paul Kavari, has done the mutational landscape for squamous cell carcinoma. So we put this together, and you can make some resequencing beads that can actually sequence each clone. And we know that every induced pluripotent cell clone comes from a single cell that was reprogrammed. And so in the population of your skin, if you go out, I know everyone says in Seattle that it rains all the time, but the only times I've ever been here, it's been beautiful. I'd be out in the sun all the time, and you get these mutations that actually are pre-malignant. So if you take a bunch of iPS clones and then do this resequencing for mutations, you can then basically do this, where you can then look at the list of mutations that are known to be associated with squamous cell or basal cell carcinoma. And then soon we'll know more for Merkel cell as well, from work that Paul's been doing. But you can then look at the different clones here and then pick out the ones that in the corrected iPS cells have the least mutational burden. We call this just cleansing them of bad genetic mutations. And then you have a defined cell bank that's autologous, that's corrected, that has a low probability of causing cancer. Because you'd hate to basically say, Mrs. Jones, we just cured your EB, but we've given you squamous cell carcinoma. That doesn't really go over very well. Okay, so now the hard part is to take these corrected mutations and then make skin grafts. This is the hard issue. And so what can we do? What we need is a way of taking, and the idea here is that you have these iPS cells that have their chromatin that's open. That's the instructions of making every tissue in the body is available to the cell. That's what an iPS cell is. But a keratinocyte, in terms of chromatin origami, has folded its chromatin in ways that the only thing that's accessible is the instructions to make a keratinocyte or the instructions to make a liver cell or a muscle cell. And the rest of the instructions are closed in heterochromatin. And so the hard part here, when you take these iPS cells to make keratinocytes, is the art of, I call chromatin origami, which is to how you fold it in a way that then only instructions that are available are to make the end tissue that you want. And this gets back to what Paul was saying about how do you control these cells. If you want to use cells as drugs, you have to then understand how they're going to act. And then you have to understand how they're going to act by defining their chromatin, how the chromatin will operate in different microenvironments. And if we can control that, then we can actually use cells as drugs very effectively. And that's really the purpose. And so really think of it like MasterChef. you're trying to find the recipe that then allow the chromatin to fold in the right way so that you make keratinocytes. Okay, so we've developed such a protocol, a 60-day protocol, which is now feeder-free, xeno-free, defined. 60 days can make go from either ES cells or induced pluripotent cells and go through, actually recapitulate human development in a dish. It's actually quite beautiful. As a developmental biologist, to watch them spontaneously develop into keratinocytes in a dish, where they look just like human keratinocytes, where they express keratin 14 and the lineage selector P63. These are keratinocytes down below, and these are induced keratinocyte-derived iPS cells. And then here you can see how we define them using FACS analysis, where the pluripotent marker OCT4 is gone, both in keratinocytes and in these iPS-derived keratinocytes. And they have nice keratin 14 in blue and keratin 18 in red. So these are the markers we use for our release criterion to make keratinocytes. So that's nice. And you can take these keratinocytes and then make grafts on the back of mice that look like beautiful human skin. And so this is really exciting for us, that you can actually, and more importantly, because we've corrected the genetic defect, in this case for collagen 7, now they make beautiful collagen 7 in green here in the basement membrane zone. And as you know, mice are not sleepy. They're running around banging themselves on their cage and fighting with each other. And these grafts survive 4 to 6 weeks in that hostile environment. So that's actually really great preclinical data that they're sticking on with pretty good glue. Okay, so this then suggests that therapeutic reprogramming can be used for correcting an iPS cell and making all sorts of different tissues, including this, what we're calling this mature skin product. What are the issues that remain that we can talk about in the discussion to think about for moving this to the clinic? Okay, one question which actually was the focus of the International Summit on Human Gene Editing, the U.S. National Academy a year and a half ago, is how much of the genome should we be editing and in which cells? So this is actually a whole new era of the ability to take human stem cells and then do genome editing. Okay, so we're taking a devastating monogenic disorder like epidermolysis bullosa, correcting the defect that they started life out with, making it just basically like a normal person. But with this technology and the ability to regenerate tissues, you can actually take a normal person and make them better. And so where are we going to stop? Where's the ethical line in terms of the combination of stem cell biology, genome editing, and tissue engineering? So for example, now we've sequenced almost every organism in nature. We know that like cheetah muscle is very fast twitching. And if you want, for example, a very fast athlete, maybe genome edit their muscle and make cheetah muscle myosin, for example. I'm not that tall. I'd like to be taller. Maybe a few more vertebrae. Whatever it takes. Like maybe we get giraffe DNA, and I can be really tall. But those are the kind of questions that we need to think about moving forward, and is already on the radar for the U.S. National Academy. But for us in the audience, eventually it's going to come down to is how do we use genome editing, stem cell biology, and tissue technology, and where do we stop? Okay, second is safety. So one of the things that the FDA is really crazy about is safety. And if you manufacture, like any manufacturing protocol, if you manufacture a tissue from autologous iPS cells, how do you know that thing you're putting on the patient is safe? What about unincorporated embryonic stem cells that then can make teratomas or convert into something else? And that's a big issue. And so we try to address this by basically taking embryonic stem cells and asking, if we just mix 50-50 mature keratinocytes derived from the embryonic stem cells and the embryonic stem cells themselves, shake them up and then put them onto the graft of a mouse, can the embryonic stem cells actually incorporate into the skin? And that's this experiment here, where you can see that you can get beautiful GFP-labeled iPS or ES-derived keratinocytes will beautifully incorporate into skin. even clones here, you get clonal expansion. So you're getting the stem cell at the basal layer making clones if it's maturely derived from the embryonic stem cells. What about the embryonic stem cells themselves? And so far, just to let you know, we've done probably 40 experiments looking to see if an embryonic stem cell could actually incorporate into the skin, and the answer so far is no. So that the issue really is that the keratinocytes have a very adhesive environment that like clicks, they stick together and they exclude a lot of foreigners. And so embryonic stem cells don't have the adhesive ability to actually integrate into the keratinocyte. And so that's actually a big advantage. Also, the keratinocyte media that we use is selective for keratinocytes and actually kills off almost all undifferentiated cells. So that's actually a really big advantage. So I think in terms of safety, we're doing, knock on wood, doing pretty well. The big issue really now is efficacy. And that is because we know from different induced pluripotent cell lines that for the same patient, you make 10 clones and correct them, the efficiency of making downstream tissues is actually wildly variable. That's a huge issue. And so the question is, how do we overcome that? And to do that, what we really need is a Google map. We need a map of basically how to get from Menlo Park to San Francisco or from Tacoma to Seattle. So how do we get, what's the map, the chromatin dynamic map from start to finish? Because if you want a self-driving car, you need to have the map. You need to have a map of how the chromatin folds, and then basically, the starting point, you pick the cells that are at the right starting point, and the end point, and you can make them go. And that's really, the focus of my lab really is focusing on chromatin dynamics from start to finish, and making a chromatin map of every place where the chromatin goes from start to finish. And so this is really a wonderful project from this talented postdoc, Lin J. Lee, who basically used multidimensional genomics, techniques which basically look at the expression genome-wide and the chromatin accessibility genome-wide, the chromatin conformation genome-wide, and all the transcription factors genome-wide, and look at how it works. Really to look at, in this Waddington model here, how the ball rolls downhill and finds the right valley, if you will. And that's not to, but what we can see is, if you look at this is RNA expression genome-wide called RNA-seq, and you can see that the pluripotency genes will turn off, like OCT4. The simple epithelial genes like keratin 8 will come on, and then as the cells convert from a simple epithelium to a stratified epithelium, it'll turn off. And then the mature keratinocyte genes come on. You can watch this happen over 60 days in a dish. And so it's a beautiful model system to watch human skin development in a dish, but also you can actually then look and develop this genome map, this chromatin map, which then it can actually give you and tell you not only the cell identity, but how it could move in a different environment. And that's really, again, going back to using cells as drugs. As Paul mentioned in the beginning, we want to know, if we want to squirt something on a patient, we want to know how that thing is going to work. With small molecules, we know that if the pH is right and the PK is right, we know the biodistribution in an animal by pharmacology. We know how that small molecule is going to work in a patient. Is it once a day or twice a day? Does it partition in the brain or not? We know about that for small molecules. For cells, we need to know the same thing. If it's in this environment, how long is it going to last? The cells have infinite amount of capacity, have an infinite amount of functions, but also can change and morph in a wide variety of ways. And that's really the challenge we have for using cells as drugs. Okay, so just to summarize, this is not surgical grand rounds. This is not medical grand rounds. We're not going to have regenerative medicine grand rounds to try to figure out how to use cells as tissues. And so far, we're trying to take two distinctive approaches. The scalable ex vivo correction, which I talked about, which is the iPS-derived cell-based therapy for therapeutic reprogramming. The more parsimonious one is direct in vivo correction using genome editing technologies, which, again, is close but not there yet. So hopefully, the combination will be very effective. And with that, I'll just stop and wanted to thank the groups at Stanford, a wide variety of patient groups, NIH funding, California Institute for Regenerative Medicine, and Family Foundation funding, which has really been instrumental in hoping to try to develop a therapy like this. Not 18 years, but maybe 8 or 4 would be really great. So thanks. I'll take any questions.